Over the last few years, there's been a lot of interest in treating patients that have so-called DNA repair deficiencies, particularly patients with BRCA1 or 2 mutations. And a class of drugs called PARP inhibitors has been investigated initially in the laboratory and has now moved into the clinic. These drugs are very effective against BRCA-related cancers in the laboratory, but as we brought them into the clinic, we see that their effectiveness is not as great, but it still clearly has a role. Olaparib, also known as Linspara, has been approved for ovarian cancer, and so have other PARP inhibitors. In breast cancer, there are no approvals. These trials have been ongoing, and two key trials were presented at this meeting. The first one is Olympia that I won't discuss, but I want to focus on another one with a drug called Talazoparib. This is one of the PARP inhibitors that is probably the most active. And the way we describe PARP inhibitor activity is by the degree to which it traps the enzyme and allows it from repairing DNA. So-called PARP trapping activity, and of the different PARP inhibitors out there, that is the one that has the highest PARP trapping activity. So not only is it potentially the most effective, but it's also difficult to combine these drugs with chemotherapy. There are some PARP inhibitors like Veliparib that are being combined with chemotherapy. So this study, the ABRAZO study, was one that was performed in patients who had multiple treatments for BRCA-related cancer. So all these patients did have either a BRCA1 or 2 mutation. The results of the study showed that there is activity. About a third of patients responded, a little bit lower than what was presented in the phase 1 study, which was published last year. This was a study that included all different tumor types, and among the breast cancer patients, the response rate was 50%. But in this study, it was around in the mid-30s, and the duration of the responses, however, was brief, 4 to 5 months. So a little bit disappointing in the duration of the responses. And this is also what we're seeing in ovarian cancer. In fact, in ovarian cancer, it's very interesting. If you sample some of these patients' tumors, you will see that the BRCA mutation has actually reverted back to the wild type. And what this tells us is that this is a genetically unstable group of tumors because BRCA, of course, encodes a DNA repair gene. So these tumors tend to be hypermutated. They tend to change their DNA more frequently. So the likelihood that there may be 1 in a billion cells that happens to have resistance through an additional mutation is quite high. And over time, of course, that clone will be selected for, including further mutations that actually changes the mutation back to wild type. So we have to learn more about how DNA repairs itself. There are other parts of the DNA repair pathway other than PARP that can be targeted. So the future of this field is really going to be to understand the basic biology better and to be able to subvert resistance.